Thanks
Okay,
for
well, thank you very much for the invite. And what I'm going to walk you through is really an encapsulation of some research we've been doing over the past decade in Australia to try and understand what might happen if bee numbers dwindle and what the implications are. Australia actually happens to be a very interesting case study. So I'm going to walk you through some of what we've found out. I'd like to also acknowledge some of my co-authors, so Javier Garcia, Manny Shastra, Scarlett Howard, and Alan Doran. And there's actually many others who contribute to this all around the world, and you'll see their names in references I put up. So to give some background sort of context, uh, about 2008, 2009, the world started to properly quantify what bees might be worth to the modern economy. And that was because Varroa destructor had started to have a big impact in California, in the United States of America. And they had noticed there'd been about a $1 billion US drop in farm gate profits. And so this went to White House level, and they started to fund some professional economists and ecologists and teams to try and quantify what this was worth and what the implications were. In 2011, a report came out of the United Nations Food and Agriculture Organization, which was a bit of a meta-analysis and has been well supported by subsequent studies showing that bee pollination or insect pollination, but predominantly bees, is responsible for about 1 in 3 bites of food we eat and is worth well north of 200 billion US a year. There's equivalent sort of values for scale in Australia. So this is a big problem which has sort of landed on people's doorsteps. I'm very much in this talk going to be looking at an ecological side of the question and how that informs some of our decision making. We know there's been a lot of discussion in the past decade about bee declines, the insect apocalypse, and this has resulted in art movements around the world. For example, the top left image shows a spontaneous piece of street art done by Louis Matty. When you go, we're taking you with us, sort of representation that bees are important. The other icons we're seeing here are various street art, or actually the figure in part D is the Eureka Tower in Melbourne. And there the architect wanted to really convey the importance of bees and how they also work in harmony as part of a city in a major installation of his artwork on Eureka Tower. We know that humans' relationship with bees is ancient. The first depictions of bees goes back to cave art over 8,000 years ago. And in what's easily accessible, this is an article which we have coming out next month in Beecraft, where we looked at ancient representations of bees on coins and right through to modern times. So some of the very first coins ever made represented bees, and that was fought, and this is a theory we've just published, that was fought because we valued honey and used honey as a tradable commodity before we actually had money. And then when we had money, putting a bee on it stamped authority that this is something actually important to own, something quite valuable. Another image you might see on that Beecraft cover is the new £1 coin, which has just come out in England, which represents a couple of bees. And that was personally promoted by King Charles III as part of a sustainability initiative he has to try and improve our understanding of bees and their impact in the world. Okay, so I'm very much going to start to unpack the work we've been doing to understand what the story is in Australia and around the world, and especially what it might mean for the ecology of the planet. So, if we go back to Victorian times, it was kind of assumed that there was something very special about humans. Look at the size of our brain, and we see colour, and that's something very special to us, and all other animals must see just black and white. And Lord Rayleigh, famous from Rayleigh's scatter, his discovery of Rayleigh's scatter, wrote a paper in Nature, very much inspired by some of Darwin's writings, going, well, actually, we see flowers and lots of colours. Maybe it's a little bit of a strange perspective that that's for only for us. Maybe animals also see colour. And we know from our visual perspective, when we go from a black and white to a colour image, it becomes much easier to see the flowers. So going back to the black and white image, we can see a few flowers, but most are very hard to understand or find. And as soon as we switch on colour, it becomes a much easier visual problem to solve. And of course, bees are interested in collecting nutrition for themselves. And as a result of their visits to flowers, the flowers get cross-pollination and have become angiosperms, or flowering plants, have become the most dominant land species of plants. Okay, so this talk's going to be jumping around a little bit because we're going to be talking plant side and we're going to be talking bee side. And actually, the interrelationship between this is what's really important. So, very much in the light of what Lord Rayleigh had written, a young Carl von Frisch in 1914 conducted a study, a very clever study, where he provided drops of sucrose on a blue square, which was surrounded by lots of grey squares. And these positions were randomly shuffled, and all the cards were covered with glass, so the bees couldn't scent mark. And you can probably see there's some little watch glasses, and you'd provide sucrose in that. And what Carl von Frisch proved was the bees could always find the blue square, and so they could not be using intensity, they actually had colour vision. Carl von Frisch went on to win the Nobel Prize in 1973 for his life's work on bees and revealing what important animal they are to human society. We've also done a lot of work in understanding bees' spatial vision. So colour vision is one part of our visual system of an animal. Spatial vision is like when we go to the optometrist and can we read a chart? And because bees have a compound eye, their acuity is relatively poor. So we can see in the panel of figures from A to F here, a flower moving from about 5 centimetres away through to about 50 centimetres away in figure F, and it largely disappears. So bees need to be fairly close to flowers to see them, and colour is really important to the bees being able to find a good feed. So it's just good to keep that in mind, and I'll go into more detail about bee colour vision in a minute. This figure here is in human colour vision, so how we see the world. So these are our colour categories. And this is a large study we just published a couple of years ago, where we looked at the possible effect of either biotic pollination, that is, where flowers are pollinated by an animal, for example, insects or bees, birds, or they're wind pollinated, which is abiotic pollination. And what you can see in the two sets of histograms, and if you think about the blue one, when there's biotic pollination, we get many blue flowers, we get many pink flowers. When there is no animal involved, we don't get any blue flowers. And so we know that animal vision is driving the evolutionary process of how flower colours work. That's human side, and I'll go to animal side now. If you look at the left-hand figure of the plots of the photoreceptors, what you'll see on the x-axis or the base of a figure is wavelengths. And I know we've got a couple of physicists in the audience. Wavelength is a quantitative way of describing the colour of light. It also describes the energy of light. So from about 400 nanometers through to 650 nanometers describes a human visual spectrum. And you can see ostensibly Newton's rainbow above the plots going from blue through to red. And when in the 1964, the colour photoreceptors for bees were first measured, and here we see data for honeybees and bumblebees, it was observed that they have an ultraviolet photoreceptor, a blue and a green sensitive photoreceptor, but they do not have a red sensitive photoreceptor. And so above that figure, we see a photograph of a yellow flower. And if we re-photograph that through an ultraviolet sensing camera, we see it goes from being a plain yellow colour to the petals are reflecting ultraviolet and the centre of the flower is absorbing ultraviolet. And then we can do a false colour representation to show for our visual system how this is a two-coloured flower and has quite salient signals or obvious signals for bees. On the far right-hand side, we see the colour photoreceptors for humans up the top left, figure A. Then we go to bats, which actually are colourblind, so they don't see colour. They only have one type of receptor. Mice and most mammals are only dichromatic. Bees have three types of receptor. When we go right down the bottom, we see birds actually have four types of photoreceptor, including a red. So they have very different way of seeing the world to what is our perspective. And when as a young man, I learnt this and I was taking photographs of flowers, it really blew my mind that because once we understand how different animals see the world differently, it gives us some insight into our own perspectives. At first, this might seem a little bit of a controversial point, but we've all probably done what's called an Ishihara color test when we went for our driving license. And that's because about 3% of a male Caucasian population only has dichromatic color vision. Some people call it color blindness, but that's not correct. It's just a different color visual system. And so even within humans, we know there's color variability. And some work shows that women actually, because of the way the opsins express color receptors, may have tetrachromatic color vision. And that's probably why we're also good at arguing about what color to paint our room, because we have slightly different color visual systems. So animals have different visual systems to us. So when we want to talk about the color of flowers, we have to get a little bit more quantitative about what this means. So here we see a representation of a bluish flower, which are honeybees visiting, or a red grevillea, Australian native. These are both Australian native plant flowers. And the blue flower can be described by this spectral chart on the wavelength scale, and this is a relative amount of light being reflected. And we can see the red flower reflects very, very little, not very much short wavelength radiation, but above about 550 and especially above 600 nanometers reflects a lot more radiation. You'll also see some horizontal arrows there. These are called marker points. These are where there's a steep change in the reflectance curve, and these become very important for some quantitative analyses to look at different visual systems. I know I've given a lot of technical data there, but this will start to make it easier to understand the slides which are to follow. Okay, this is a map of inside the bee's brain. So we're quite lucky because Carl von Frisch had won the Nobel Prize, particularly in Germany and in Europe. There were a lot of leading groups which went through to looking in detail how the brain processes information. And we know that bees have a very sophisticated color visual system. And there are some analogies to how our brain manages color. For example, and this is for the experts in the audience, I know there's a couple. We have the M&P pathway, which control our color and our spatial vision. We see similar processes happening in the bee, and this tells us a lot about the commonality of how a visual system evolves. This slide shows actually data for a bumblebee. This is very much in the light of what Carl von Frisch showed us about how bees can be trained and conditioned. And what we see is the response time of bees relative to their accuracy at choosing between two very similar hues of blue. And we see that some bees, so on the bottom left of the lower plot, are very fast but are quite inaccurate. And some bees take more time and are more accurate. But then we play a funny trick on the bees. We make any of their errors associated with a punishment of a bitter tasting quinine. Now, these are free-flying bees. They elect to participate in the experiment. So if they don't like it, they can literally buzz off. But they continue to play in our experiment, but they all improve their accuracy. And this is evidence, and this is why we published it in Nature, this is evidence of volitional control in bees. They are making decisions about what they want to do and how they prioritize information. This becomes important for bridging across to the next slide, or the next couple of slides. When I showed you a map of a bee's brain, when we use psychophysics of how the bees actually perceive colour, we can build colour models of how their brain represents different flower colours. So if we just look at the top left-hand figure, which is the Australian case study, you'll see a colour hexagon with terms like UV, that stands for UV photoreceptor, up the top blue, and the far right green, and then different colour categories. This is a mathematical interpretation of how they're perceiving colours, and the white dots show the loci of where flower colours sit. And this gives us a representation of how their brain is perceiving the information. Now, this might seem very complex, but actually any of you who are very au fait at calibrating your colour monitor, we use a CIE colour space, and it's a mathematical representation so that when I wrote this talk for this evening and I chose some colours, they're turned into numbers and they are sent by the internet and they turn up in your computer and they represent correctly. And that's also using a colour space. So this is a very typical tool used for colour management, and some very accurate ones have been designed for bees. Okay, the top left-hand figure here we have seen before, and that's the colour photoreceptors for honeybees and bumblebees, together with the visual spectrum for humans. But what you'll see is now I've added to it a purple camel hump, which sits over the top. And that's a psychophysics. So psychophysics means We have got information from the bees by doing behavioral experiments with them, and we know how they process the information. And it shows at 400 nanometers, they are fairly accurate at discriminating wavelengths of light or colors, and 500, they're even better. But less than 350, they're not very good. And at 450, they're also, it's not their best. So this is called the von Helderson psychophysics function for the honeybee. Why this is important is when we look at our right-hand figure, the top part of that shows us some typical flower reflectances. And I mentioned the marker points before. And so we, for each flower reflectance, can quantify a couple of numbers which describe its color. And on the bottom part of that, we see a set of histograms, which is a stacked representation of all the flowers which have evolved in the Northern Hemisphere, actually in the Israel region of the Northern Hemisphere. And we see that around 400 nanometers and 500 nanometers are the most frequent flower colors. And we again see placed over that the von Helderson color discrimination function for honeybees. And so this was published by Lars Schicker and Randolph Menzel in 1992 and was taken as potential evidence of coevolution between bees and flowers. But then Lars Schicker did a phylogenetic analysis, and he showed that the visual system of honeybees, this is what we're seeing in the top left-hand slide, is phylogenetically ancient. So the ancestors of bees had exactly the same color photoreceptors before flowers ever evolved. So it's been a one-sided story, but flower colors have evolved to suit the color visual system of bees for bee-pollinated flowers, because I'm going to show you some different examples in a minute. As mentioned before, we can plot all these in a color space and we can get a relative representation. And here we can see that most flower colors which have evolved for in what's called the blue-green region of color space, where there's very few flowers which are pure ultraviolet in color. And part of the quest we have followed for the past decade is understanding this evolutionary process. Okay, so I read a lot of this background work during my doctorate about 20 years ago in Australia, and I thought this is all fantastic theory the Europeans have developed, but I'm in a land down under, as I imagine most of us in this talk are. And what's the story in Australia? And Australia is interesting because we have a significant sea barrier from about 34 million years ago. So potentially flower colours may have evolved very differently in Australia. So I worked with Steve McLaughlin, who's a paleobotanist, and could look at the fossil records for flowers. And we also, with Mani Shastra, an expert botanist, measured enormous numbers of flowers in Australia. And that's what we see here in the main plot. The top right-hand plot is what I showed you a minute ago for the Northern Hemisphere. And so what we saw was parallel evolution of flower colours around the world. Exactly the same process has happened in Australia as has happened in the Northern Hemisphere. What we do see in both data sets here is some surprising data at 600 nanometers, so which is not explained by the von Helderson function. And so we spent a lot of time trying to understand that, and we're able to solve that by, again, this is a brilliant work of Mani Shastra. He was able to look and identify either bee-pollinated or bird-pollinated flowers, look at their phylogeny, and what we saw in the previous slide at 600 nanometers, is red flowers to our eye, which are bird pollinated. And so these birds entered the pollination story many tens of millions of years after bees, and flowers quickly evolved to, if they liked being, or if they were optimally pollinated by birds, they quickly evolved long wavelength reflecting signals. And here, this is just a representation that we can develop quite sophisticated mathematical models to represent the colour vision of birds. And what's particularly important here is this red arm, which we see this is strong group of loci of flowers which have evolved to bird vision. Okay, so I was very interested, or our team was very interested in what this might mean for climate change. So I've been going slow, giving lots of definitions. We're going to speed up now. So the first thing we did was look in New Zealand. There were reports in New Zealand that going up to the height of mountains, maybe 2,200 meters, there were much fewer bees and many more flies. And we went and we measured the flower colors there, and we mapped them to the von Halvorsen function, and we actually found, oh, it's about the same data set. They're bee pollinated. But we did not give up that easily. I convinced Manish Shastra, most amazing guy, as Australian citizen, and very, very proud to be a very good friend of his. But he's originally from Nepal. He won many prizes to come to Australia and do his doctorate with me at Monash University. And that was important because we decided to do a study in the Himalayas. And the conversation went something like this. Manny, do you think you could have a backpack and carry a computer and a spectrophotometer and a car battery to power it? And the top right-hand figure shows the terrain he was climbing up through. And he climbed up to well above 3,600 meters, up to over 4,000 meters, and measured all the flower colors. And we were quite surprised that we saw very similar distribution of the histograms shown in the bottom right-hand figure here, which is also the signature of bee pollination. But he did observe, and his colleagues in Nepal also observed, bumblebees forage up to over 4,000 meters. And so that explains this. So we literally scratched our heads and went, well, what can we do to understand? And this time we're looking for very global effects as we're refining some of our computer models. What can we do to find a place where there's no bees? And actually, I'll come to that in a minute. So whilst we're in the mountains, we also got contacted, while we're doing mountain studies, we also got contacted by this fantastic group in Taiwan. And Taiwan was a very interesting island for us to look at because you go from sea level to well above 3,000 meters in a very short distance. And so we again convinced this wonderful guy, Ken Chung Tai, to carry a spectrophotometer up to quite decent heights and measure all the flower colors with a spectrophotometer and build phylogenies of how these flowers had evolved. And we saw globally that there was a fairly similar distribution to Australia, actually a study which had been done in Japan, and also the study we'd done in Nepal. We saw in Taiwan, as we went from low to mid to high altitude, however, something was happening. The data was getting more concentrated, and that was a study we published in 2021. And then on here's just a representation of how these colours sit in for the different altitudes, some low, middle, and high altitudes relative to the data I showed you for Australia before. So we're seeing the main peak is similar, but something is changing. And then a study which we just published 2 weeks ago, also in Frontiers in Plant Sciences, showed that actually, as we change altitude, there is a change in the ratio of the spectral signature, so the color signature, relative to the achromatic contrast of the flowers. And we also see that the flowers get smaller with higher altitude. And we think this is an interaction of different bee species. Whilst they have a phylogenetically conserved visual system, as they exist in different environments, they have slightly different sizes, and this changes how their visual system works and their acuity works. As Jenny mentioned, we're also interested in looking right back to the dawn of the first flowers. This is a study which we've also just published in the last couple of weeks in Proceedings of Royal Society. And if you look at the top left figure, we see a nice pink flower sitting against a variety of backgrounds. So we see some red rock and some grey rock. And we were quite interested in whether the ability to see signals against a background might have been the driver for ancient insect vision and why it evolved trichromacy, so having an ultraviolet blue and green photoreceptor. And we spent about 10 years doing this study. So we collected rocks from Cairns down to Wilson's Promontory or South Point and Wilson's Promontory, right across Australia. And we measured their spectra and we built computer simulations of how these applied as backgrounds for flower colours. And what you see in the top right-hand figure is the blue plot is a distribution of flower colours, which I've been showing you before, for bee pollinated. The pink plot is for bird pollinated, and especially we see the very strong peak at 600 nanometers, which is the red arm you saw before. But the shaded brown color is the distribution of background colors. And so what happened was background colors strongly have marker points at just over 400 nanometers and 500 nanometers. And ancient insects before bees evolved trichromacy to avoid flying in a complex environment and not colliding. And then when flowers came along, first to bees, the flowers evolved those signals, but they exaggerated the signals. So you can see the pink flower on the top left is a much stronger signal. So we now know why flowers evolved the colors they did. And this probably happened in the early mid-Cretaceous, when the first flowers and the first bees evolved. The co-evolution, or the evolution, because the visual system of bees is conserved, happened very, very quickly. Okay, I want to quickly go back. There's only a couple of slides on temperature and then a slide to finish up. Another study, this is a study I did at Cambridge University in the United Kingdom, and we're very interested to know what happens if a flower has signals, color signals, but it's offering a different reward because of its temperature. So if it's a cold day, we all like having a nice hot chocolate. And if it's a hot day, we like having a cool glass of water. We wanted to know, does the bee's physiology also take account of the temperature of the nectar they're receiving? And so what we showed is if we have a purple and a pink flower of the same temperature, the bees just choose at random. It's just take any nectar. However, and these experiments are done at 20 degrees ambient temperature, which is quite cool for a bee. Their thoracic temperature needs to be about 30 degrees for them to fly well, so they need to be quite warm. If we make the purple flowers warmer, the bees prefer the purple flowers. And if we make the pink flowers warmer in a crossover design, the bees prefer the pink flowers. Then in Australia, when I came back to Australia, we repeated this experiment in the top left-hand figure you see with the sugar bag bee, Tetragonula carbonaria. And we saw they also had a preference at low ambient temperatures, so 20 up to 30 degrees, they had a preference for choosing a warmer flowers. But when the ambient temperature goes above 32 degrees, there's a quick switchover in how the bees prefer. This links to the data I showed you on speed accuracy. This is volitional control on these insects. They are choosing to modulate their temperature depending upon their environmental conditions. Then with Manishastra, what you see in the bottom figures, and I don't expect you to take account of all of them, I'm just showing you diversity. We measured plant flowers, and we show that plant flowers also modulate their temperature depending upon the ambient temperature. So for some flowers, as it gets warmer, they just heat up. But other flowers, as it gets warmer, they are able to cool themselves, and they use some fairly interesting physiological principles to do this. And so this means in a scenario of climate change, there's going to be some winners and losers. Some plants will adapt very well to the scenario and some won't. And the bees are quite sensitive to this system. And we have preliminary maps suggestive that in different parts of Australia, some of these different plant species are being more or less successful. The full map of how that is going to unfold with climate change is unknown, but we've got at least a footing on understanding the dynamics of the system. Okay, I'm going to jump back these last few slides to the quest we had of a world without bees. What happens if our bee numbers drop? And we were able to locate this island, Macquarie Island, deep in the Southern Ocean, which for some nice coincidence, no bees have ever been to. And this is a very rare situation. I showed early on in the talk that humans have a very close relationship with bees. So nearly anywhere humans go, we have taken bees with us. But no one ever took bees to Macquarie Island. There are no bird pollinators at Macquarie Island. There's seabirds, but they don't visit flowers. But there are fly pollinators. And the photographs you see in the bottom right-hand figure are photographs of the flowers from Macquarie Island, which we collected. These flowers come from a diverse phylogenetic background. And if you look at their closest ancestors, which are on mainland, they are quite colourful flowers. But when they are exclusively pollinated by flies, they quickly evolve to be a dull yellowish green colour. And we know flies have a slight preference for yellowish colours, and they have a very different way of seeing the world to bees. To put that slide I just showed you in terms of the marker points we've been discussing, the main figure shows flower colours. The main figure on the right shows the histograms I've been showing you for bee pollinated flowers at 400 and 500. The red data is from Macquarie Island. So you get a complete knockout at 400 nanometers. And for so we have to do quantitative analyses, we have to build statistical models with that data. But the figure in the bottom left, you can see it with your own eyes. Even though our own eyes aren't designed for doing quantitative analysis of colour, we can see a big change in colour. We've also very recently, this was a study led by Javier Garcia, replicated this on mainland Australia, actually near Melbourne. by looking at orchid species. And we looked at orchid species which are either bee pollinated, which is top left image, or the one next to it on the right of it is a fly pollinated orchid species. And we built these into full computer models and looked at the phylogeny of how this had transpired. And if you knock bees out of a system, you knock all colour out of a system, and very quickly. So we know that bees are super important to the ecology of flowering plants. Okay, this is my very final slide. So what we're trying to do at Monash University, and this is out of Alan Doran's lab, is, and this is some wonderful work by a doctorate student who's just received his doctorate actually. We're building computer systems which can track in agricultural scenarios how bees fly and land on plants. So this is the individual tracks in the main figure of bees as they're flying and landing. And we've been able to quantify when at a farm or an agricultural site, when a certain plant species like strawberries has been successfully pollinated. And this means as we try, because we've got several different things we're trying to understand, the ecology, but that from all the work we've been looking at, we've also trying to understand how we're going to feed a population going forward. So we're trying to develop smart agriculture. So if bee numbers drop, we can actually try and use them in a much more intelligent way or smart way so we can keep our food supply up. And so that's an overview of the work of several labs I work with over the past decade. And I hope it's generating a few questions.
Okay.
Jenny, there is a question to start off with
Yes.
on the chat already. Not about bees actually, but the question is from observation: why do cabbage white butterflies appear to love the colour purple, as you find in lavender?
Yeah, it's a really beautiful question because my wife asked her the other day.
I see.
We have many cabbage butterflies around at the moment, and we also have lavender in the front garden. She goes, and actually it was usually the bees or over the lavender, but we've seen very few bees this year, and this is just our front garden, and heaps of cabbage butterflies. And she asked me why, and I had to answer, I actually don't know. As I showed early on in the talk, we're very fortunate with a model I presented that Carl von Frisch revealed how you can train bees and get high quality data. When it comes to most other animals, we just don't have experimental access, so we don't understand how they perceive the world. So it's a beautiful question, but we don't have any data to inform an answer. I'm suspicious. Well, I can give some educated guesses. So work by a Japanese group, Kentaro Arikawa's group, shows that butterflies, and this is Japanese butterflies, actually have greater than 5 different types of colour photoreceptors. So they have a richer colour perception than birds. So we are suspicious that moths and butterflies might have an even richer colour sense than birds or we do, but the psychophysics is lacking. That's so you can measure the colour photoreceptors, which is an electrophysiological measurement, but to actually understand what an animal's doing, you have to literally do a behavioural or psychophysics experiment, and I don't think anything's known for cabbage butterflies.
Right. Okay.
Psychophysics is a new topic for me. I'd have to say the idea of psychophysics is quite an extending one. Adrian, if you were able to stop sharing your screen, we might be able to go back to the gallery view of people, and then people could put their hands up, such as Robert. But while he's chewing, I might take the liberty of asking a question of my own, which is, I was reading a a short paper in the New Scientist, which is saying that air pollution is changing the scent of flowers and confusing insects, that the air pollution is affecting the scent range, and so they don't know quite what to do. They've got sensory pollution, as it were.
Yes, this is a big issue. There's a group at Melbourne, within Australia, there's a group at Melbourne University who's done some work on that, and there's several groups around the world. Ostensibly, and I'm probably giving a low-level answer because it's not my expertise, but the pollution clogs the receptors. And when you clog receptors, they don't work very well. And this can become quite an issue for finding flowers. So as I showed early on in the talk, their acuity is quite poor. The best way to envisage how an insect might be seeing their world, if you think about when we go swimming at the ocean and we don't have goggles on, what our acuities are like for seeing underwater, that's something like what an insect's seeing. So we can certainly see our friend at like 1 meter or maybe half a meter, and we might make out, if I'm quite close, you can make out I'm holding up a hand. But as I go further away, everything just evaporates because our visual system is not tuned to that aquatic environment where a fish sees very well in that environment. So because their acuity is quite poor, they actually do rely on scent quite a lot for orientation. So we think scent is a very big context setting cue. Insects going along will detect some scent. We know the colour vision is very important for finding a flower, but it needs a context of, smell something, now I start looking carefully. And this might even be analogous to if we're out walking somewhere and we smell some nice food, we go, oh, must be something nice around here, and we look. And so it's a big problem.
Thank you. Robert, I'd like to unmute and ask your question.
Yeah, thank you, Adrian. That was a really interesting talk. I went along to a talk by Ken Walker, who's the...
Yeah, I know Ken. He's a great guy.
the conservator of insects at the museum. He was saying there's about 1,700 species of Australian bees, and about a third of them, I think, just from memory, are short-tongued bees. They go out to flowers with short corollas, and another 2/3 of them are long-tongued bees. I was wondering if any of your team has done work to see whether the Australian bees, have obviously been separate from honeybees for millions of years, have a very similar colour spectrum.
Yes, so this question unpacks a few different ways. For the colour photoreceptors, we know that's phylogenetically ancient. It actually hasn't been measured for Australian bees, but we have strong enough data from around the world to know that they will have a UV, a blue, and a green photoreceptor, very much like the number of humans for whom we have ever measured colour vision is extremely small, but we can build very, very good computer models which explains colour to us, like what's transmitting the colour now. However, that's at receptor level. Then I showed one slide of inside the bee's brain. Then there's lots of neurons which are processing that colour. And there is data suggestive that those neurons can weight the signals in different ways, which gives rise to colour preferences. And we know in humans, we have a variety of different colour preferences. The majority of humans prefer blue, but there's certainly some humans who like yellow or pink or purple. And the reasons for that are often very personal on that, but it's probably something to do with how our visual system's wired up. So when we went and measured the colour preferences of bees, so for the honeybee, Apis mellifera, they have a strong blue preference. So does Bombus terrestris. In Australia, we measured the sugar bag bee, so it's a stingless bee, Tetragonia carbonaria. It has very similar colour preferences to Apis mellifera, but it's separated by about over 70 million years on the evolutionary scale. So we think it's fairly phylogenetically conserved. However, there is one study done by Scarlett Howard which tested a couple of Australian native bees and showed some slightly different data. So there's an urgent need to do more experiments, but each experiment takes years and there's not a whole heap of people lining up to fund the work. So we try and target very specific species. So the sugar bag bee is known to be an important pollinator, so it was quite an important one to test early. It would be fantastic if people did short-tongue and long-tongue bees. There's just too little known.
Thank you.
So
Well,
we kind of know an answer, but we don't know in detail.
it raises an interesting question, Adrian, that you've just touched on about, we know this is an important topic. How well are we kind of tuning into it, and how well are the people who you rely on to support the work that you feel we need to do, how well are they kind of being responding to the need? You know, the insect crisis, the threat of extinction, as it were.
So internationally, there's been a strong response, especially in Europe. And so there's some reasonably good support mechanisms have emerged. Australia is a long way behind the eight ball. Look, without putting with no political flavour, there was certainly one side of politics which anything to do with climate change was not a high priority. There was another side of politics who was aware of some of the issues with bees and had some conversations pre-pandemic and had some plans, I think, which is the current government. I'm not sure if it's on their agenda at the moment because there's lots of financial issues and
These are down the list.
Look, I'm certainly not a member of government, so I don't know all their priorities. But it's a challenging time in the world.
Steve, would you like to unmute?
Yes. Still on colour perception, am I to understand that bees generally don't have a red receptor? Does that mean that they are not interested in bright red roses?
So you're quite correct. So no bee which has ever been measured has a red photoreceptor. So they will not see red as red. Now they can still find red flowers, a bit like you don't need to see, if it was lower light here, you wouldn't need to see colour in my hand to know this is a hand against a light background. So if they fly down low, they can see the shape of a flower against a skylight, but they would see it as a dark shape against a bright skylight. They will find red roses, but many red flowers, which you see bees on, they're actually nectar robbing. They're not pollinators. So one of the main hypotheses for the evolution of red flowers, this is a good friend of mine, Klaus Lunau's hypothesis, is called the bee avoidance hypothesis. And so bees can actually do damage to flowers if they don't enter them correctly. And so if you decide you want to be pollinated, well, when I say decide, it's an evolutionary process, it's not a cognitive process. But if a strategy has evolved to be pollinated by a larger vertebrate, so a flying bird, then that species of plant might do well to avoid colours which the bees like. But the bees are only interested in getting nectar. They're not being altruistic of trying to pollinate for pollination services. As Darwin very clearly pointed out, each species is acting in its own interests. And so if they can nectar rob, they will.
Okay. Before we go to Robert, I wondered if you might just go, if I could take you back to what I read in the abstract of the paper that is published in the Royal Society at the moment. We say we present tangible solutions for how technological applications such as mining of information from social network sites and the use of imaging data combined with artificial intelligence can enhance our understanding of pollinator networks. And I wrote that in the publicity. So
Yes,
I'm wondering, I'm wondering if you could just briefly say, how does I was intrigued really, how does social media networks come into it? And what's the role of AI? Because that's another thing we need to try and get our heads around.
Yeah, so it's a great question. I pulled a few of those slides out because I timed my talk beforehand and it was going to be too long, I thought. But so it's a great question. So we worked, this is in Alan Doran's lab. We worked with some very brilliant PhD students and researchers. And the problem we were trying to address is, how do we get data across Australia where, like Mani Shastra has to go out and spend 6 months measuring all the flower colours and trying to log what's there in one site? How can we get a view of what's happening across all Australia? That's logistically impossible. And we brainstormed this beautiful solution where, well, we can use incidental citizen science. So there are many people who love bees and flowers and are going out, taking photographs and uploading them. And so what we do is then mine that from Flickr, for example, and then use a reverse image search to validate it. We also did on a subsection some hand validation, and we looked at a couple of key model species for which there was high quality data, and we showed in maps that we could replicate for a couple of key species with social media images what was the known ranges of those plant species. And then just last year, we published a study actually from Japan, which was very informative. Japan was a really interesting case study for us because of the cherry blossoms. They had historical written records going back many, many hundreds of years, ostensibly 1,000 years. The Japanese tourists and many people who go to Japan are also avid adapters of new technology. And so they photograph everything now. And if you, so it's a bit divergent to what I was talking about, but if you went and looked in The Conversation, we wrote an article about that. Do you know The Conversation?
Go, sorry, you go.
Just for the general audience, The Conversation is an online dissemination for academics. We wrote an article, and it's got a beautiful video within it, where we could see going up Japan the signature of when the plants were coming into flower by when images were being uploaded by people. So we could actually we could capture the warming of the seasons just by how many images are being captured. And that's just not something you could do with researchers and clipboards. because you need literally thousands of observers, and so social media is giving us way new insights or fantastic new insights on the process. And so it's a combination of a very detailed studies, like I showed you Monash Shastra had done, and then, but detailed studies cost a lot of money, take a long time. They tell us one part of the story very well, and we combine that with some of the social media data, and we can make inferences to understand the overall process.
Thank you. And the cost is low as well.
Yes, it's a trade-off of big N. You don't have the same precision, but you get a very different understanding of the story, and very much like the Australian government might do in a census statistical analyses of where do we need schools, etc., etc. And then in another study might want to do something very deep, an ethnographic study on what type of people need what type of food or uniforms or that, but you can't do that for everyone.
Robert's got his hand up, and I'm conscious of time. So if anyone else is sitting on a question, so to speak, can you please indicate, because I think we'll have to finish shortly. So Robert, would you like to unmute and ask your question?
Yeah, Adrian, you were talking about bees as nectar robbers. Some 30 years ago, I went on an Earthwatch project with David Payton on Kangaroo Island. He was studying a comparison between the behaviour of feral honeybees and honey eaters. And what he found by excluding bees or honeybees or birds from particular trees was virtually all the trees were cross-pollinated, which takes a really big animal to need to move to several trees. And the bees were getting up actually earlier in the morning than the birds. And by the time the birds got up, most of the nectar was gone, and they were essentially robbers. And his study ended up convincing the national park managers in virtually every state to exclude feral honeybee hives from national parks, kill them, because they're actually quite damaging to the lives and ecology of honey eaters.
Yeah, there's a fantastic researcher in Australia, Kit Prendergast, who I've collaborated with, and she's done a lot of work on Australian native bees, and is quite vocal and has good empirical data to show that in some ecological settings, Apis mellifera is actually causing more of a problem to the environment than it is beneficial. Of course, Apis mellifera is so important agriculturally, we have to manage that issue, but there does need to be a lot more work on native species to understand the implications of how we use Apis mellifera.
That's
well, not all bees are all bees. There's differences, like there's diversity in humans and how we have opinions. Different bees and different insects do things in different ways.
He was just looking at Apis mellifera.
Yeah.
Saw that as a really serious problem in national parks.
Yes.
The problem is there's a lot of farms adjacent to national parks. It's very hard to stop the bees getting in.
Yes. Yeah. Well, Kit.
One of those diabolical problems.
Well, I don't disagree with you. I try and avoid emotive words like diabolical. It's certainly a problem which needs management, and it's way under researched of how to do it well. And as I mentioned, Kit Prendergast has been quite vocal that we just need to understand native bees well.
Thank you.
okay. Any last questions? Anyone? Okay. Adrian, you've given us a lot to think about, and I'm sure you've enlarged our perspective about bees. It is a huge topic, and one that we're all struggling to get our heads around, especially as the climate changes and, what does the future hold. So we're very grateful for you sharing your expertise and time with us this evening. So if you could all join with me in thanking Adrian in the usual fashion.
Thank you for your time and great questions.
Thank you, Adrian. Really good talk.